Hi sir, we are going to report about um, techniques in biotechnology. Then our assigned report is plant transformation. So what is plant transformation? It's a way to insert DNA from another organism, normally another plant, into the genome of a plant of interest. So definition of plant transformation. So we have the transformation. It refers to the introduction of a gene of a choice into the genome of organism. These genes of one species can be modified, or genes can be transplanted from one species to another. Most transgenic organisms are generated in the laboratory for research purposes. So plant transformation is to insert DNA from another organism, normally another plant, into the genome of a plant of interest. And currently, Acrobacterium-mediated transformation is the most commonly used method for plant genetic engineering because of relatively high efficiency. Plant transformation consists of 3 steps. We have target genes, plant tissues, and transformation method. Let's discuss about what is target genes. So target genes have a vector, plasmid, and plasmid vector. So vector is one of the most important element in recombinant DNA or rDNA technology and in gene cloning. So a vector should have the following features: contain a replicon that enable it to replicate in host cells. It should have several marker genes. It should have a unique cleavage site within one of the marker genes. So the insertion of foreign DNA into the marker genes leads to an inactivation and identification of recombinant molecule. For the expression on clone DNA, the vector DNA should contain suitable control elements such as promoters, terminator, and ribosome binding sites. So plasmid is a small cellular double-stranded DNA molecule that is distinct from a cell's chromosomal DNA. Plasmid naturally exists in bacteria cells, and they also occur in some eukaryotes. So plasmid have a double-stranded closed cellular DNA molecules which exist in the cells as extra chromosomal units, are self-replicating, found in a variety of bacterial cells as accessory genetic units, advanced growth to the bacterial host. Lastly, plasmid vector. Scientists have taken advantage of plasmid to use them as a tools to clone, transfer, and manipulate genes. Plasmid vectors, it should contain an origin of replication that operates in the organism into which the clone DNA is to be introduced. So let's have our next reporter.
Plant tissue cloning in plants. Plant tissue culture for transformation. First, a plant part is cultured. Next, callus grows. Next, shoots development. Next, for the shoot development is the shoots are rooted plants grows to maturity. Plant tissues is a morphogenetically component cells in plant transformation, morphogenetic process that lead to plant regeneration. Plants are multicellular eukaryotes with tissue system made of various cells type that carry out specific functions. Muna siya ang plant tissues. Then dirita sa first stage, a plant part is cultured. Muna siya. This is a tissue culture involves the use of small pieces of a plant tissue or the explant, which are cultured in nutrient medium under sterile conditions using the appropriate growing conditions for each explant types. can be induced to rapidly or produce new shoots, and with the addition of suitable hormones for the new roots. That is a plant part is cultured. A plant part is kanang kuan siya, a cells of an explant or a tissue. Ano siya, ang a plant part is cultured. A material siguro siya. Callus growth. Calluses are the body's ways of protecting the skin from excess pressure and friction. So as long as these conditions exist, the calluses will continue to return. Ang callus growth kuno daw siya ang body's way of protecting the skin from the excess pressure and friction. Siya ang kuan lugar, guard, kasangga. So as long as these conditions exist, the calluses will continue to return on ang callus grow. Next is the shoots development. The new growth from seed germination that grows upward is a shoot where leaves will develop. As, as shoots grow and age, the cells develops secondary cell walls that have a soft structure. Some plants produce toxins that make their shoots inedible or less palatable. Ang shoots development, this is the stage where a shoot where leaves will develop as shoots grow and age. Mura na siya, paingon na siya sa pagtubo na siya dahon na ni. Next is the shoots are rooted, plants grow to maturity. Mura na siyang last stage na sa pag-culture, sa tissue culture. The roots grows downward in the direction of gravity, which is positive gravitropism, and the shoots grows upward away from gravity, which is negative gravitropism. Gravitropism. The reason plants know which way to grow in response to gravity due to ameloplast in the plants. Namugoy kini siya nga stage, dili yan tumatagan kung straight ba yung pagtubo or sa kilid ba, upward or downward, due to gravitropism, which is undenied. Thank you.
Is the transformation methods or DNA delivery. So, when we say transformation methods, this is the process by which an organism acquires exogenous DNA. So, in DNA delivery, the most common technique for direct transformation is the microprojectile or particle bombardment. So, this is a technique based on the use of particle gun or gene gun.
The device used for bombardment can act on any type of cell, transforming not only the nucleus but also all the cellular organelles. Then, ang microprojectile bombardment is usa ka technique which foreign substances such as DNA are introduced directly into the living cells and tissues into the plants.
So, when we say protoplast polyethylene glycol method, is that muna siya ang first technique to report the successful integration of foreign genes into a plant cell. Protoplast electroporation method. So, this is an intensive electrical field that leads to pores on the plasma membrane and allowing the DNA to enter. Silicon whiskers. This is a silicon carbide fibers to punch holes through cultured plant cells. A silicon carbide fibers and cultured plant cells are added to a tube and vortex vigorously. The mechanical force generated by the vortex drives the fibers into the cell. Indirect method agrobacterium. So, this is a pathogenic soil bacteria that contain a TI or tumor-inducing or RI, which is root-inducing plasmid. Agrobacterium tumefaciens. This has been extensively used as major agent for the production of transgenic plants in a wide variety of plant species. A tumor characteristics may change often abruptly growth rate.
Histological structure, invasiveness, or responsiveness to extraneous stimuli such as hormones or chemotherapeutic agents. These characters tend to be independently variable and subject to independent progression. tDNA is an expression of genes located in the tDNA transforms, the host cell in a tumor cell resulting in a tumor crown gall formation. So, because the tDNA transfer system of A to mutations is the most common DNA delivery tool for the genetic engineering of the plants.